Welcome to my video. This video is about cap stage, which is the third part of tooth development series videos which we are doing. In our previous video, we saw how a tooth bud developed, right? And in this video, we are going to see about cap stage. So basically, the enamel organ, which is in the shape of a bud, that is a flower bud, is changing into a shape of a cap, like a head cap. The tooth germ, which is in the form of a flower bud, logically, when the cells multiply rapidly, it should grow into a more larger and more spherical tooth bud, right? Like this. But it doesn't happen. Instead, here unequal multiplication of cells takes place. That is, unequal growth takes place in different parts of the tooth bud, and this leads to the formation of a cap shape of the enamel organ. And this cap is characterized by a shallow invagination on the deep surface of the enamel organ. Now, let's see the parts of the enamel organ at the cap stage. In bud stage, we only had two types of cells, right? The peripheral low columnar cells and the centrally located polygonal cells. In the cap stage, the peripherally located cells of the tooth bud changes into outer enamel epithelium and inner enamel epithelium. That is, the low columnar cells seen in the bud stage differentiates into outer and inner enamel epithelium. And the centrally located polygonal cells changes into stellate reticulum. And this is dental papilla, which is seen in the invagination of the cap. And this is dental sac. The peripheral cells covering the convexity of the cap are cuboidal cells. These are cuboidal cells, alright? These cells covering the convexity of the cap, which are cuboidal, are called outer enamel epithelium. The cells in the concavity becomes the tall columnar cells. This is how tall columnar cells look. The cells which are low columnar in bud stage changes into tall columnar cells, alright? And these tall columnar cells in the invagination are called inner enamel epithelium. And this inner enamel epithelium, that is the tall columnar cells, will further develop into enamel producing cells, that is ameloblast, in the future stages. And the inner enamel epithelium is separated from the dental papilla and the outer enamel epithelium from the dental sac by a membrane called basement membrane. This is called stellate reticulum. Now let's see how the stellate reticulum is formed. At the early period of cap stage, the polygonal cells are located at the center of the enamel organ. These polygonal cells secretes glycosaminoglycans, which is seen in ground substance. These glycosaminoglycans, that is GAG, has the capacity to trap water, alright? That is, they are hydrophilic because they attract sodium and potassium ions. This brings in water, alright? Let's say these are the polygonal cells, alright? These cells secretes glycosaminoglycans in the intercellular spaces. Now, these glycosaminoglycans secreted from the polygonal cells exerts osmotic force, and so the water is drawn into the enamel organ from the dental papilla or the ectomesenchyme. Now, what happens is the water which is getting into the enamel organ from the dental papilla pushes the cells away from each other since they are present in the intercellular spaces and causes the cells to become star-shaped. But they still remain in contact with each other by cytoplasmic processes. This is how the stellate reticulum is formed, alright? The main function of the stellate reticulum is that it acts like a cushion and a shock absorber. That is, it has a cushion-like consistency as it is filled with fluids. So, it acts like a shock absorber by distributing the shock to protect the delicate enamel-forming cells, that is, the ameloblasts. Now, let's see about transitory structures. These are temporary structures that disappears before the enamel formation. The cells in the center of the enamel organ are densely packed and form the enamel knot. This knot projects in the part near the dental papilla at the center of the invagination, showing a knob-like structure. Alright? At the same time, a vertical extension of enamel knot occurs, and this is called enamel cord. This enamel cord extends to meet the outer enamel epithelium and divides the stellate reticulum into two parts. That's why this is called enamel septum. At the point of meeting of the enamel cord to the outer enamel epithelium, it shows a small depression. This is called as enamel navel because it resembles like an umbilicus. The main function of enamel knot and the enamel cord is that it acts as a reservoir or storage of the dividing cells for the growing enamel organ. Now, let's see about dental papilla. This inner enamel epithelium proliferates and influences the underlying ectomesenchyme, that is, the ectomesenchyme enclosed by the inner enamel epithelium. And so, this causes proliferation of the ectomesenchymal cells, and the ectomesenchyme condenses to form dental papilla. Alright? This dental papilla is the formative organ of dentin and primordium of pulp. That is, the dental papilla changes into pulp and the cells adjacent to the inner enamel epithelium changes into odontoblast, which produces dentin. This condensation of dental papilla is not just the crowding of cells, but blood capillaries are also seen. That is, active budding of capillaries takes place and mitosis is also seen. That is, new blood vessels are formed and multiplication of cells takes place. And the cells of the dental papilla present peripherally adjacent to the inner enamel epithelium enlarges and later differentiates into odontoblast, which produces dentin. Alright? Now let's see about dental sac. While the enamel organ and the dental papilla develops, the marginal condensation of the ectomesenchyme surrounding this enamel organ and dental papilla takes place and forms a denser and more fibrous layer and forms the primitive dental sac. Now let's see about enamel niche. The enamel organ may have a double attachment of dental lamina to the overlying oral epithelium, enclosing the ectomesenchyme. This is just a histologic structure seen under microscope. This double attachment of the dental lamina to the oral epithelium is called enamel niche. This appearance is due to the funnel-shaped depression of the dental lamina. Thank you for watching. If you like my video, do subscribe, like and share. Have a good day.